Thank you, Phil. So I did realize yesterday that 10 years ago was my first BBS conference. 10 years ago. At that point, I just started working with Phil. And many things have changed now. We are all older a little bit. But and many things have changed in the way we understand Bardet-Biedl syndrome and in the way that we are approaching therapies. So today, I'm going to try to give you some details about the research, perhaps at some point a little bit too much technical. So if you feel that I'm overdoing it, just let me know, say boring, and then I will stop. But I do really think that some of them, they are interesting, and I think that you will appreciate what we're trying to do if you understand that point. So let's start with gene therapy. Gene therapy is a way to replace the genes that all you have mutated. The reason why you have Bardet-Biedl syndrome is because you have those genes that are not working anymore. So the easy idea is, what if we can deliver to those cells that are not able to use your own BBS genes with a normal copy of the BBS genes? Will these cells be able to function? Nay, here we go. Third time lucky. So this is a really old slide from Phil. Probably he was 12 when he put this slide together. And it's just a summary about the syndromes that you have in BBS. There I have a small graph of all the genes that so far we know they are mutated in Bardet-Biedl syndrome. And it's just to show you that the most important ones are BBS1, which is the gene that most of you have mutated, and the second one is BBS10. And I'm pointing down to blindness and obesity. And that's because in our animal models, those two features are the ones that are always found in all the mutant mice that we produce. So far, in all these years, we've been able to produce, we and other labs, to produce different mouse models where the different BBS genes are mutated. And we were lucky enough to find that all of them have a similar retinal degeneration than the one that most of you suffer. And on top of that, most of them, all of them, displays an obesity phenotype, and they all put weight in a similar manner that you also do. And that's really helpful because if you want to try to rescue some of these organs, you need to have a model that have a similar phenotype than the one you have. So. So far, and that's something that we started 3.5, 4 years ago without knowing anything about gene therapy because we were not experts in gene therapy. We started in a project where we were trying to use these techniques in our mouse models to see if we were able to repair the cells and the tissues that are damaged. And we focused in two different ways to deliver the genes. One, to cure the blindness, the photoreceptor degeneration that I'm going to talk a little bit now, and the other one, the obesity, that we know now it's driven by a malfunction of a neuronal network that is at the base of your brain, an area that is called the hypothalamus, and that is regulating the function of one of these hormones that Jeremy was talking before, which is an hormone called leptin, an hormone that regulates the exercise and the appetite that you have. And it seems that this whole network is not working in BBS, and this is possibly one of the reasons why you sometimes find this urge to constantly eat and constantly be picking up. So it's not easy to deliver a normal copy of a gene into a specific cell type because DNA what is going on? So DNA cannot go through the system and enter in the cells because the cells have this membrane that protect them. So we need to find a vehicle, a bus, a train to put our DNA inside and move it into the cells. And luckily we have virus that their function is just to do that, to go into the cells, deliver the DNA, and stay there. So what we managed, we didn't, but in the past many groups managed to use different kinds of virus, remove everything that was not necessary, and just keep the vehicle in order for us to be allowed to put this gene inside. And we are using one of these viruses that is called AAV, adeno-associated virus, doesn't matter. And we put in there our BBS constructs, and then we produce this virus, and we give this virus to our animal models. That's not working. They were wrong. So all the main group of this study is done in one specific gene, which is BBS1, because it's the most common one. Which is BBS1. And in order to do that, we use a mouse model that have the most common mutation for BBS1 and the most common mutation with BBS, which is the M390R. We are lucky enough that the genes of the human BBS1s are very similar to the genes of the mouse BBS1. This way, if we change just one small amino acid, a small bit of these genes in the mice, we have the same phenotype that we have in humans. So the first idea is, can we stop, halt, delay the retinal degeneration? And in order to do that, we need to know a little bit about what's the eye and what's a retina, which is the layer of cells that you have behind the eye, and are the ones that are touched by the light and send the electric message to the brain that make us all see. And in BBS, the cells that are dying in the retina are these cells that are called photoreceptors, which are these specific ones at the back of the retina. And we think that those are the cells that we need to target. Those are the cells that we need to give normal copy of BBS1 in order to stop them dying. There are two types of cells, of photoreceptors. One are the we call rods, which are the ones that are working with there is not much light, and the other ones that are called cones, which are the ones that work better with colors and with a lot of light. Both of them are dying. But then there are other cell layers in the retina, and we don't know if we are going to need to tag them. Like the layer at the top, the black one that you see at the top of these sections, which is called retinal pigmented epithelium, and that Helen probably is going to talk to you later on. So two things that we need to consider. First, what type of cells are we attacking? And the second one is when. Because if we go really early, we are going to be able to stop the degeneration of these cells for a longer time. And then we are going to be able to keep the vision for a long time. Type of cells and when. So we know that BBS1 is expressed in these photoreceptors. So we designed a construct that was specifically targeting these cells. And we put the human BBS1, your human BBS1, into one virus and injected this virus subretinally. under the retina. So the virus could spread around the retina and then infect all the cells. But then, how are we going to try to understand if we are rescuing the vision of the animals? Obviously, we cannot ask mice, what kind of letter are you seeing there? So in order to do that, we have to develop what is called an electroretinogram. Doesn't anybody of you have an electroretinogram done before? So what you usually do is just have some electrodes put in your eye, and they are flashing you different light intensities. And this way, we can record the electricity going through your brain. And we know if these photoreceptors, these cells, are working. And we know if the animals can see. In order to do that, we have a really small machine with some electrodes that are 2-3 millimeters. And we have to put these electrodes in the eyes of the mice. And we have to do that in the dark, completely darkness. And you can see Jasmine back in there. Jasmine, where are you, Jasmine? Jasmine is one of the members of the team. She is down there, working with a BBS one animal just before we are going to do this analysis. And this way, we can detect if the rods, which are these cells that detect dim light when you are in the dark, or the cones, which are these cells that can detect colors, are being rescued. We can differentiate between both of them. I'm going to jump that. So we did that in animals that were 1 month old. That means that these animals are starting losing vision. We inject the virus subretinally, and we follow the vision of these animals for a whole year. And what we saw is that animals that were injected were able to keep the vision, and the animals, the mutant animals that were not injected, obviously were not able to keep the visions. And that worked for both kinds of cells. And just to give you an idea of the level of rescue that we found in these animals, they are keeping 50% of the vision that a normal wild-type mouse will keep, which for a mouse is quite a lot, quite a lot. That means that we can sustain the vision of these animals if they still have some photoreceptors. But we were not happy with that because it was only 50%. So we create another virus. And this time, what we were doing is we were targeting not just the photoreceptors, but also the cells in this retinal pigment and epithelium that is also important for keeping the cells alive. And we are doing that. We're injecting these cells when the animals are 1 month old. And we are also doing something else, which is injecting really, really, really small animals when they are 7 or 8 days. They are really tiny pups, and we are able to inject our virus there. And so far, we have a good response, and the injected animals are keeping the vision much better than the untreated ones. And the second project is related with the obesity, the way you put weight. And in the last 2 years, there were a few publications that were relating these problems with the incapacity of the hypothalamic network that I told you before to read the signals of this hormone. It's called leptin. So what we did is just, okay, can we make these cells restore the function of BBS1s in these cells, and then can we monitor the weight of the animals? We changed a little bit our construct because obviously it's not the same an eye or a brain. And we injected P0 animals, that means newborn animals in the brain with our virus. And we followed them for a year. Another treatment that we use as well is instead of doing our intracranial injections, doing intravenous injections. We did that to try to find out if we were able to target all the tissues in the body. That's something really difficult because every specific vector, every specific virus have the tendency to go to one place, one organ, one tissue. And we were trying to do that, trying to target the eyes, the brain, the kidney, and the liver, and so on, and so forth. So we create a new constructs, we inject the animals. We saw that we were able to see the human BBS1 that we were injecting after a year of injection, so that's really good. That means we can keep the expression of this new gene that is now the one that has to take control of the function of BBS1 for a whole year. And what do you think happened there? After a year, our animals didn't put any weight. Especially the males, they behave better than the females, you know. And now we are analyzing these results. When we did the same experiment, but injecting intravenously, we saw an improvement. These animals were lighter, both males and females, but statistically, the difference was not completely significant. And we are trying to improve that now. But still, really encouraging that we can find a difference between treated and untreated animals. So what are we doing now, and what will we do in the future? The first thing is that we did create a new BBS sequence that works better than the wild-type one, the one that people without mutations in BBS1 have. There are ways to do that, and this sequence works much better. So we create this sequence, and we create new virus, and right now we injected, again, all of our animals, and we are measuring the weight of these animals to see if we are able to improve our results. And another thing that we are doing is that we did another construct with a new gene, BBS10. So BBS10 now is playing into the equation as well. We have the animals ready, and we are going to start injecting them with the new virus and see if we can rescue the weight and other phenotypes. So the question is that we are doing really well. We can rescue the vision of our mice, but we still are on the way to move that to a clinical trial. Clinical trial meaning to try that in human beings. Right now, we cannot cure BBS, but I'm absolutely convinced that we are in the right way, and in the future, and I don't know how many years that could be, but not many years. I think that we will be able to start trying some of these therapies in you. And finally, this work has been, for the last 4 years, has involved many people, and I would like to thank especially the people that have been working directly hands-on. So Monica and Sandra and Jesmi and Mira. You have Jesmi and Mira here today, so please go and ask questions to them. Here we go. And obviously, Phil Bills and other researchers like Professor Robin Ali, James Braithwaite that you had here 2 years ago, Ahad Rahim and Simon Wellington that are missing from this slide. So thank you very much.
What we're going to do is go through all the talks, and then we'll have a group session in terms of questions.